Welcome, everyone. Very excited to be with my new friends, Chidosi and Linta, this evening. My name is Julia Brisky. Most of you know, but if you don't, I do the International Co-op Program at IHN. I've been here 17 years, and I do the programming for our continuing education and am faculty for professional practice. But that is not why we're here. We are here to see and hear the amazing Dr. Chidosi and Linta give us some information on Vitract. Now, they're going to answer all your questions about that at the end. They know everything about it. Chidosi is the CSO, Chief Scientific Officer, and Linta is the CEO. So both of these individuals will answer everything you need to know. So for tonight, please again put all of your questions in the Q&A. I'm going to do a brief intro of Chidosi. We're going to do, and Linta. We're going to then do the 1 hour talk at 45 minutes, 45 minutes an hour. Okay, then we're going into the Q&A and then we're going to have the giveaway. So tonight, Dr. Ojobor will start with key gut concepts, then explain various lab techniques for identifying and measuring microbial levels in the gut. A focus will be on the role of gut dysbiosis in chronic inflammatory diseases. Remember, he will be coming from the perspective of all of his education and, of course, Vitract, which is Linta and Chidosi's lab testing facility for digestive health. Really interesting stuff. Linta then is going to wrap up by outlining the various players in microbiome testing and offering guidance on choosing the right tools for different client types. Thank you, both of you, for being here this evening. I'm looking forward to hearing about everything you have to say. And please introduce yourselves and then roll right in for us.
Thank you, Julia. So I'll take it first before popping it on over to Dr. Ojobor. We're really excited to be here today. Vitract started it really rooted from my own personal experience. When I was in university, I started experiencing all this inflammation in the joints of my hands. It came out of nowhere. I went to go see the doctor and eventually found my way to a great integrative care provider where I worked with her for a little over 5 months with the focus of healing the gut, and that worked for me. I reversed all my inflammation, got off all my medication. But the one thing that really stuck out to me was not only all the care out of pocket, but all of the supplements and the different protocols and the different testing, everything was out of pocket. And I had a really hard time accessing the test at the time, which I remember paying $600 out of pocket for a gut test. And that stuck with me. And it wasn't until years later that I met Dr. Ojobor and we bonded over this shared experience of how expensive sort of gut testing is in today's world. And we saw an opportunity to do something there. So we've been building for the past couple of years, and I finally launched this test to the Canadian market. We're getting a lot of interest, and there's actually a lot of IHN grads that we worked with. So we're really excited to be here today and meet more of you. We also have our launch event in Toronto on May 10th. So we're really excited to meet a lot of you in person for the first time. And yeah, I'll pass it over to Chidosi now.
Yeah, thank you very much, Linta. And thank you very, very much, Julia, for having us. I'm very excited to be here. Excited because, we've been in conversation for a while, and we've reached out to a lot of, we're currently working with a lot of IHN graduates, and they're fantastic. But also excited because we're going to talk about the gut microbiome. And personally, my academic training started with virus research. I was very interested in viruses that cause gastroenteritis in children. But prior to that, I had done a lot of bacterial research work. So I considered that to be my first love. So when I was going back to do my PhD at the University of Toronto, I had the opportunity to reunite with bacterial research. If anyone is here and has done bacterial research before, you know that one of the lovely things about them is that they grow so fast. So if you mess anything up in the lab, you can literally just go back the next day and set up your experiment again. During my PhD, I was developing these novel therapeutic tools to target disease-causing bacteria, especially the antibiotic-resistant bacteria. And I was actually using viruses to do that. I was genetically engineering viruses that can't infect human beings, but can infect bacteria. I was genetically engineering them to kill bacteria that would not that were resistant to antibiotics. It was in the middle of that I got really fascinated about gut microbiome because this was a high precision tool that could manipulate complex biological ecosystem, and that's what one of the things that spiraled into this. Linta and I met, and we thought that we had to innovate within the space to solve not just the problem of providing a comprehensive report, but also an affordable test for people. So I'll just go straight to share my screen and then talking more about the test. And like Julia said, if you have any questions, please feel free to put them in the Q&A. Can everyone see my screen?
Yep, we're all good.
Fantastic. Okay, so today I'm going to be talking about, I want to focus on the role of gut dysbiosis in the establishment of disease. And if you think about it, conversely, it's pretty much the role of eubiosis, that is the opposite of dysbiosis in the establishment of health. And one of the some of the things that we're going to look into very quickly today is first the spatial biogeography of the gut microbiota. I'll basically be talking about the different localization of bacteria across your gastrointestinal tract, why that is important when you're thinking about health and disease. I'm also going to touch on the concept of gut dysbiosis. This is a terminology that is being thrown, almost everywhere now, but what exactly is it? And how can a practitioner actually truly understand the concept in the context of the symptoms that the patients are having? And then I'll also touch on the key component of a 16S microbiome test. I'm going to talk a little bit more about the 16S later into the talk. But lastly, I'll wrap up by showing you a real life case study of a patient who works with one of the clinicians that's using our test. And I will show you how, the clinicians are able to glean more insight into their gut health by pretty much just looking at, their gut microbiome profile. Now, when you talk about the gut microbiome, one of the things that is very, very core to the understanding of the gut microbiome is the way these different microorganisms in your gastrointestinal tract are localized in different anatomical sites. Now, this happens because there are several conditions that actually sets the stage, for that. Now, as you're going from your esophagus down to your colon, one of the things that you see is that you're seeing different conditions change. As those conditions change, they come with some organisms being enriched and some organisms losing that diversity in that particular anatomical site. As you go from your esophagus down to your colon, you can see here that there is an oxygen gradient. Somewhere in the stomach, you still have the partial pressure of oxygen to be much higher than when you're going down to your colon. In fact, when you get to your gut lumen, what you tend to observe there is what we call the hypoxic state. That is, very, very almost zero amount of oxygen. The implication of that is that you're going to enrich anaerobes, strict anaerobes. And these strict anaerobes, are usually associated with health. In fact, a lot of research that has gone into truly understanding gut dysbiosis would tell you, it's called the oxygen hypothesis, would tell you that when you're looking at the gut lumen, when the gut intestinal barrier is compromised, that can actually lead to the leakage of oxygen into the gut lumen, and that can set the stage for facultative anaerobes, which are these other organisms that can thrive in the presence of oxygen. Unfortunately, those organisms are the organisms that are usually pathogenic or that can cause disease. So you can see that oxygen, the gradients there is determining the type of organisms that would be enriched in that anatomical site. Here you can see that in the stomach, aerobes such as Lactobacillus prevotella, you can see some amount of of those aerobes in the stomach region. In the small intestine, we tend to have more facultative anaerobes versus in the gut lumen where you have strict aerobes. But it's not just oxygen that determines the variability in that bacterial localization in the different anatomical sites. It is other things like your bile acids. Your bile acids, which are produced by your gallbladder, they will be released for lipid metabolism. One of the things you see is that in the small intestine, you see that the acids themselves tend to acidify that environment, and that also enhances organisms that can thrive in that particular microenvironment. So is the case when you're looking at short-chain fatty acids. When you get to the colon, we know that short-chain fatty acids are pretty much, products of, fermentation of dietary fiber. And all of those things, the purpose of all of those things is that all of these, conditions and variables come together to help set the stage for one, the bacterial composition that in that microenvironment, but also secondly, what they would do, because it's not just about who's there, it's also about what they're doing. Anything that would distort that balance is what we call gut dysbiosis. And so when you talk about gut dysbiosis, what exactly are we talking about? When I talk about gut dysbiosis, you're trying to capture the terminology in three facets. One, you're talking about the imbalance in the bacterial composition. Now here, when you talk about bacterial composition, you're looking at the richness of the gut as well as the evenness of the gut. What do I mean by that? The richness is you're looking at the total number of species in your intestinal tract, which gives you a sense of the bacterial diversity, but also it's not enough for your gut ecosystem to be rich. They have to be evenly spread out, because if two individuals, for example, have 500 species, but if for individual A, two species actually make up 90% of the 500, we cannot exactly say that that gut is evenly spread out. So you want to get a sense of, that balance in bacterial composition. And you also want to get a sense of, what the balance is between the beneficial organisms, the probiotic bacteria, the commensals, and the pathobionts. I'm going to go into those definitions very shortly. The other thing you want to look at are the changes in the bacterial metabolic activity. So now you're looking beyond, just the taxonomic profile of the bacteria. You're looking beyond, bacteria A, B, C is there. Now you're talking in the context of the functional profiling. What does this bacteria do when they're there? Are they producing beneficial metabolites? And if they're producing these beneficial metabolites, what are they? Are they producing short-chain fatty acids like your butyrate, your acetate, your propionate? We all know about, the role of things like butyrate cannot be overemphasized. They're the primary sources of energy to your colonocytes. They help to contribute to the maintenance of your intestinal barrier. They play high anti-inflammatory roles. They are involved in, insulin sensitivity, name it. In my opinion, I think that that's one of the most important biomarkers of health. And then lastly, you also want to think about the changes in the bacterial localization. And the reason I'm spending some time to talk about gut dysbiosis here is that the ViTrac test is a test that gives you a sense of gut dysbiosis. And if there is anybody who truly wants to understand the microbiome on a gut-wide scale, you understand that the root cause of almost every disease, starting from cardiovascular health to digestive health to metabolic diseases, all of them have ties to the gut. And it is about dysbiosis. Now, one of the biggest conundrums in the space is how to actually truly define what a healthy microbiome is. And what I mean by that is that, there's been a lot of studies that have gone into this for over 15 to 20 years. And the goal has been, how do we look at someone's gut microbiome profile and say that it is healthy versus it is unhealthy? Now, that definition is a little bit hard to come by, but what is not hard to come by are the microbial signatures that actually define a healthy gut from a dysbiotic gut. So in a healthy gut, we tend to see high bacterial diversity, richness, and evenness. So high number of species that are evenly spread out. We also tend to see the type of organisms that are known to produce beneficial metabolites. I had just talked about the short-chain fatty acids, but it's not just the short-chain fatty acids. You also have things like the neurotransmitters. You know that over 90% of the serotonin that is produced in your body is actually produced in the gut, but it's not just serotonin, it's dopamine, it's GABA, it's acetylcholine, it's all of those neurotransmitters. There are organisms, the bacteria resident in your intestinal tract that are known to produce all of these organisms. Now, it's also not just neurotransmitters, it's also about the B vitamins vitamins, tons of bacteria that are producing these beneficial metabolites. Now, and then in a healthy gut, one of the things that we know marks a healthy gut is low levels of pathobionts. Now, what are these pathobionts? The first place. Now, pathobionts are part of your intestinal flora. When they exist in small amounts, they do not cause harm to you. But when the condition is set right, where they can overgrow, then they can actually portend the establishment or the progression of disease. And some of these pathobionts, the way they do that is they produce toxins or like endotoxins like lipopolysaccharides, which is actually part of their outer structure. We'll talk a little bit more about that. Or they produce gases that can actually contribute to the to the establishment or the progression of disease. And then you also want to see a dominance of probiotic bacteria and commensals. And the opposite of that is what you see in a dysbiotic gut. Now, if you want to think about this more pictorially, this slide basically gives you a sense of what a healthy gut looks like. Now, a healthy gut, in a healthy colon, what you're looking at here is you have your epithelium, you have your immune cells, and your commensal microbes working collaboratively to establish what we call a stable equilibrium. And it is that stable equilibrium that actually preserves your intestinal health and protects your gut from external threats. And so here you're seeing the tight junctions. I'm pretty sure a lot of you have heard of things like zonulin and occludins, all of those tight junctions that are holding your epithelial cells together and are preventing microbes from translocating from your gut lumen to the other side of the epithelium beneath your lymphoid tissues, that is your lamina propria. Now, that is one of the first lines of defense. The other line of defense that you have here is your mucous membrane. Your mucous membrane is helping to maintain the integrity of your gut barrier. But it also performs another interesting function. It harbors tons of microorganisms. Now, there's tons of bacteria in your mucous membrane, and what are they doing there? They're helping to maintain the tight seal, and they're also helping to fend off these pathogenic organisms. They are also helping to establish an anti-inflammatory state within your gut ecosystem. Now, such that when a pathogenic organism, gets into that microenvironment, you find things like your innate immune cells, your dendritic cells, capturing them, sending signals to your adaptive immune system, and like your T cells, and they are producing, producing this inflammatory response to it. That inflammatory response has to be controlled. So if you have a healthy gut, what happens is that your commensals actually play a huge role in controlling that anti-inflammation, such that there is what we call the pro-inflammatory and anti-inflammatory balance. Now, when you're looking at the gut ecosystem of, people, for example, who have ulcerative colitis, well, we know that there is substantial evidence in academic literature that they experience, a gut barrier breakdown. What tends to happen is that this gut barrier is then disrupted, and then you tend to see all of these commensals that are harbored in the gut, in the mucous membrane, you see them translocate. When they translocate from the mucous membrane to the other side, they make direct contact with your immune cells. That's triggers a very complex inflammatory process. Now, because you do not have the right balance and composition of bacteria, you cannot then establish an anti-inflammatory state, and that leads to severe flare-ups, which can further lead to pancolitis. Unfortunately, we know in academic literature that extensive forms of pancolitis can actually increase the tendency to develop colorectal cancer by 15-fold. So you see, it comes from having a barrier breakdown to having chronic, inflammation, because you have that, uncontrolled pro-inflammatory process. And then that could also set the stage for almost everything when they get to circulation: metabolic health disease, cardiovascular health problem, colorectal cancer, name it. Okay, now, I was just talking about how the gut microbiome plays a role in almost everything, and everything ties back to gut dysbiosis, right? Great. Now that we have tried to give a sense of what gut dysbiosis is, we have to think about it in the context of the imbalance in bacterial composition, also in the context of the changes in the changes that we see at the metabolic level, metabolic activity level, that is the functional profile, and now the translocation of bacteria from where they ought to be to where they ought not to be, which can drive a very, very complex inflammatory reaction. I want to introduce to you the Vital Gut Microbiome Test. But before I talk about the test proper, What does this test do? Now, the VITRA test is a 16S based sequencing test that identifies bacteria and archaea based on the 16S gene of their DNA. Now, of several microorganisms from bacteria to archaea to from bacteria to archaea to viruses to protozoans and even to worms. Now, the vital microbiome test identifies the bacteria and the archaea only because these are the organisms that have the 16S gene. All right? Because these are the organisms that have the 16S gene. Now, the reason we started with this, with the 16S test, is because the bacteria makes up over 90% of your gut ecosystem. And so they're playing the hugest role in, the way your gut ecosystem is your gut, they're playing the hugest role in your gut health. And so that's why we're starting with the 16S gene, with the 16S sequencing. And every single bacteria has a 16S gene. So because they have the 16S gene, we can identify a bacteria, and that 16S gene is unique to them. We can identify a bacteria by just sequencing that 16S gene. And based off on that, we can tell you that this is bacteria A, B, C, because that 16S gene is unique to them. Okay. Next, I will show you a case study of an individual who has taken the VITRAC test. But before I show you this person's report, I'd like to establish what their symptoms were prior to the time that they took the gut microbiome test. Okay, so this is the report of an individual who took the VITRAC test. This individual suspects that they have leaky gut. They are usually chronically inflamed. One of the things that they're also experiencing is when they're inflamed, they tend to experience breakouts of acne. And this person suspects, although undiagnosed, but they suspect that they might actually have a mild colitis. And so they took the test through one of our practitioners. And so what happens is that when you do the vital microbiome test, from the time when you send your sample to a lab to the time your report will be ready, it will take about 10 to 14 days. So it's pretty much fast, and it still captures the state of your gut within the period when you're still having the symptoms that you're having. Now, in the first section of the test, you're going to see two scores. The first score is what we call the gut health score, and the second score is what we call the diversity score. Now, what is the gut health score? So pretty much everybody starts at 100. As you deviate from optimal ranges, your scores will be subtracted accordingly. So your gut health score is a percentage value of all the bacterial archaea that are listed in the test. I'll show you that shortly. As long as they're within the optimal range, you're going to be 100. For this individual, is 78, meaning that certain things are outside of the optimal ranges. If you're between 0 and 49, we consider that to be poor. 50 to 79, we consider that to be neat work. And 80 to 100, we consider that to be satisfactory. Now, the second score, which is very, very important, because this score actually gives you a true sense of your bacterial gut diversity. It is the diversity score. Now, the diversity score is computed using what we call the Shannon index. It accounts for two things. One, it accounts for the richness of your gut, and then it also of your gut microbiota, and it also accounts for the evenness. So the richness talks about the total number of species that we found, unique species that we found when we sampled your test, when we sequenced your sample. And then the evenness shows how evenly spread out they are. So, bacteria, you classify them from phylum down to class and order and family to genus and then to species. For organisms that are within a genus, they have different children, which are the species. You want that to be evenly spread out. So that captures this. So the diversity score is a very well-known biomarker for every single chronic inflammatory disorder, from Crohn's to UC to IBS, and even metabolic health disorders like insulin resistance, obesity, name it. So those are the two scores that you see in your first section. And this person seems to have 78 in their gut health score. So let's go to the second section. Now, the second section is where we actually stratify these different bacteria into health-associated bacteria and disease-associated bacteria. And I would like to spend a little bit of time here explaining this. Now, the health-associated bacteria are the probiotic bacteria and the commensals. So these are the organisms that we know in academic literature play beneficial roles to your gut health. And how do they do this? They do this either because they're helping to ferment the dietary fiber that you're taking in, and they're producing important metabolites like your short-chain fatty acids, your neurotransmitters, your longevity factors, your detoxification factors, your vitamins, etc. And commensals, they're here to help to establish that internal homeostasis within your gut microenvironment. They're establishing that anti-inflammatory state that is needed to keep that homeostasis. Now, the disease-associated bacteria, on the other hand, you can see here pathobiont overgrowth. Now, like I said, pathobionts are actually part of your normal flora. That is to say that they're actually part of you. The problem is when they overgrow. And when do they overgrow? They overgrow when you're sick, when you've taken rounds of antibiotics and it wiped out the beneficial organisms within that ecological niche. That gives rise to pathobionts to overgrow. They can produce gases. I'll show you that, and those gases can set the stage for disease. And the other categorization that we have here for disease-associated bacteria are the exogenous pathogens. So these are organisms that are not found in your intestinal flora. You picked them from your environment. A lot of people, for example, let's say you went out, you ate contaminated food, you had food poisoning, and when we sequenced your gut, we might have found things like Campylobacter jejuni, Shigella. All of those things are not supposed to be part of your gut. They're picked from the environment. Okay, so for this particular individual who is experiencing chronic inflammation, this person suspects that they have leaky gut. And this person has leaky gut. They suspect that they might have mild UC. They're usually very tired. And then when they're inflamed, they tend to have acne. We see here, these are the probiotic bacteria, Akkermansia, Bifidobacterium, Blautia faecalibacterium. You see the percentage in which they exist in this person's ecosystem, gut ecosystem. So this person, Akkermansia, makes up 0.11% of their gut ecosystem. Now, the optimal ranges that we recommend for Akkermansia here is between 1% and 5%. So you can see here that they have 10 times less than what we consider to be the optimal range. And I must mention here that these optimal ranges are coming from, most of them are coming from Dr. Jason Harlach, who is a clinician scientist. He's been doing this microbiome work for over 20 years. And we have a personal relationship with him, reached out to him, and we're adopting some of his ranges. Interestingly, those ranges coincide with the thousands of tests that we have done for people who are considered to be healthy versus people who are having symptoms. Anyways, Akkermansia here is low for this individual. Bifidobacterium here is low for this individual as well. We can see Blautia here is also low. Now, this Akkermansia, we know that Akkermansia, some of you are probably taking Akkermansia probiotics. We know that Akkermansia actually plays a huge role in, maintaining the integrity of your gut barrier. How do they do that? They help in the production. Although they feed on the mucous layer of your gut epithelium, when they feed on that, when they metabolize that mucus, what happens is that they produce short-chain fatty acids that then activates your goblet cells to produce more mucin. It activates the mucin gene so more mucin is produced, and that we know that mucin actually plays a huge role in maintaining your intestinal barrier. Some of you might have actually seen the pendulum Akkermansia probiotic now, which they call the GLP-1 probiotic. So the basis for that is that we know in academic literature that Akkermansia produces short-chain fatty acids, and these short-chain fatty acids can actually activate your L cells to produce more GLP-1, which also then activates further down the line insulin to be produced to fix glucose into cells and to be used up. So this person is low there. Bifidobacterium, they're also low. Bifidobacterium contributes to a maintenance of gut barrier integrity. Blautia, this is arguably the third largest butyrate producer in the gut. This individual is also low there. Faecalibacterium, this is the largest butyrate producer in the gut. It is the largest because it has the metabolic freedom to produce butyrate. That is to say that it can produce butyrate from different substrates. Right? It can produce butyrate by fermenting dietary fiber. It can produce butyrate from other short-chain fatty acids like acetate and propionate. We tend to see in healthy people around 10, in excess of 10%. This person has 8%. And if you go down, you can see here that Rosoboria, which is the second largest butyrate producer in the gut, short-chain fatty acid producer in the gut, this person also is low there. So there's something that is very, very consistent in their result here. It is that they lack these beneficial organisms that are known to be short-chain fatty acid producers, especially butyrate. And we know that butyrate helps to contributes to the maintenance of your gut integrity. So it's possible that that is the basis for the leaky gut, because butyrate, you do not have enough short-chain fatty acids that are contributing to the maintenance of that gut integrity. Now, so if you look at the commensal results, so these commensals, usually they are in large amounts in the gut, but we know that when they are localized in positions that they ought to be, they would help to keep an anti-inflammatory state. All right. So here we see that almost everything is optimal. Bacteroides is optimal. Allostipes is optimal. So we tend to see allostipes to be increased in people who are eating high-fat diet. And that's because they feed a lot on bile. So a lot of, as you're eating fat, bile is being produced for lipid metabolism, and that tends to enrich allostipes. And so if you look at some studies for people, for example, who are largely on carnivorous diet, we tend to see an enrichment of allostipes and bacteroides and bilophila. But they are also high in coprococcus. We know in academic literature that coprococcus plays a huge role in producing short-chain fatty acids. They're high there. It looks like all of their commensal bacteria are great. Now the pathobiome section. So here, the way to look at this report is by looking at every single section, you're getting an insight into what is high, what is low, what do they do? And that's why we provide a lot of education on top of the test. So for every clinician who is using our test, we have what we call the ViTrack microbiome, where we'll provide a lot of education on every single bacteria that actually shows up on the report. So that arms you to really understand the implication of what is high and low. Now, the pathobionts. Now, the pathobionts we sectioned into 4 categories: the inflammatory lipopolysaccharide producers. Here, proteobacteria, this person is high in proteobacteria. It has as high as 5%. We tend to see that people who go above 4% tend to have chronic inflammation. And why is that? We know that the proteobacteria have the lipopolysaccharides in their outer covering. So you look at these proteobacteria, pretty much gram-negative bacteria. You look at the outer components of these organisms, they have the lipopolysaccharides. Lipopolysaccharides is made up of lipid and polysaccharides. Now, we know from over 40 years of academic work that these lipopolysaccharides are highly, highly pro-inflammatory. Now, the problem here is when you have a leaky gut, some of these proteobacteria then translocate from your gut lumen and they start to make more direct contact with your immune cells that are beneath your lymphoid tissue. And that then sets the stage for, chronic inflammation. And we have seen this both in humans and in mouse study. So this is one of also the biomarkers that actually kind of like points to why this person might be experiencing the inflammation that they are experiencing. And I want to go back because I know that some people here are using tests like other tests where they show you things like zonulin and all those as biomarkers of inflammation. This is a biomarker of inflammation. So you have to always think about disease in terms of the host or the pathogen, because it's a host pathogen relationship. And so if you're seeing that the zonulin levels, if you are understanding inflammation from the intestinal permeability from the side of zonulin, when you're seeing high levels of zonulin, that tells you whether or not this person has inflammation. You can also learn that by just looking at the lipopolysaccharide producers, because we know that they trigger high level of inflammation and they set the stage for intestinal permeability because they break down the zonulin and all of the proteins that are holding the tight junctions of your epithelium. We also have the hydrogen sulfide producers, such as your bilophila and your Dysbiosis. Now, we know in academic literature that bilophila and Dysbiosis, these are sulfate-reducing bacteria. That is to say that they reduce they reduce sulfate to produce hydrogen sulfide. Now, the problem with that is when there is an accumulation of hydrogen sulfide in the gut, we know that hydrogen sulfide actually breaks down your mucin layer. So some of the people that we have seen taking our tests that have issues with chronic inflammation, some of them it's because they're just high in bilophila and desulfovibro, and they're just producing a lot of hydrogen sulfide, which we know breaks down your mucin layer and is a precursor for inflammation. But it's not just for people who are concerned about their digestive health. When we started using this test, one of the first set of people that actually bought into the test were people who had long COVID. And one of the debilitating symptoms that they had was brain fog. And interestingly, we actually did see that bilophila and desulfovibro was enriched in the gut ecosystem of these people. So it's possible that overgrowth of this pathobionts was actually leading to an overaccumulation of hydrogen sulfide, and that was setting the stage for, and that was seeping into circulation, and that was setting the stage for things like brain fog. The other biomarker you can get insight into are these methane producers here, this methane producing archaea. And these are not bacteria, they are archaea, so they belong to another kingdom, but they are known to actually produce, an overgrowth leads to an accumulation of methane. Now, we know that we've seen these actually in people that have chronic constipation and people that have constipation predominant IBS. And when you take a step back to think about it, it makes sense because we know that methane and a buildup of methane can actually cause the slowing of the transit time of food across your gastrointestinal tract. So this tends to be one of the biomarkers for functional constipation. Enterotoxic inflammatory pathobionts, Enterococcus faecalis, high levels of E. coli, we also know can actually set the stage for inflammation. Okay, so the last part of the section 2 are the exogenous pathogens. So these exogenous pathogens, like I said, they're not part of your normal flora. You just pick them up from the environment. Campylobacter, Shigella, Salmonella, down to Yersinia here. We did not detect any of that in this individual. Okay, so that's the section 2 of the test. Now, the section 3 of the test here, and I'm going to try to rush through this because I think I have 5 more minutes. The section 3 of the test, here we're giving insight into the metabolites. So in the section 1, I've shown you the scores for your gut health score. I've shown you the scores for your diversity score. I've shown you the scores, your diversity score. And in section 2, we've shown the different breakdown of organisms that we found. And in this section 3, what we are giving you are insight into the metabolites that all of these organisms are producing. Now, but before I go into that, I need to mention that the organisms that I showed you in section 2 were about 30 or 35. So that's just a summary of all the organisms that we know play a huge role in gut health. But one of the other things that you will get with your report is the total gut profile, every single organism that we found. Usually there could be 200, 500, depending on how robust your profile is. Okay. So section 3, I must mention here that we are not measuring the metabolite here. What we are measuring here are the organisms that actually produce this metabolite. The first is short-chain fatty acids. As you saw in section 2, this person had low levels of bacteria that actually produce the short-chain fatty acids. And that's why their scores here fall under need work. So here, green indicates satisfactory, orange indicates needs work, and red indicates high. And these scores that you're looking at here, we're basically just comparing you to other people who have taken the test. And for this individual, they fall under needs work because they just do not have sufficient bacteria from their test report that produce these beneficial metabolites. We also show information on neurotransmitter producers, so acetylcholine, dopamine, GABA, histamine, norepinephrine, and serotonin. One of the insights we also give you are the vitamin producers. We know that your gut microbiota are major sources of vitamins. And this person seems to check out in all of those. But we just don't give you insight into these beneficial things. We also give you insight into toxins. An example here is methane. Like I said, methane plays a role in constipation, cramping, abdominal distension, and bloating. A lot of people who have, issues with IBS and SIBO comorbidities, we tend to see that for people that have, the constipation predominant IBS, they tend to have high levels of methane. And so when you do your SIBO breath test, one of the things that we have seen in academic literature is for people who have these comorbid conditions, they tend to have high levels of methane in their SIBO breath test. And this is also what we see for people who have diarrhea, predominant IBS, they tend to have high levels of hydrogen sulfide if they have that comorbidity. We also give you insight into cardiovascular health. We know that trimethylamine oxide plays a huge role in cardiovascular health. Lipopolysaccharides, these are endotoxins that are highly pro-inflammatory. So you can actually, gain insight into that by looking at this. Other biomarkers we give you insight into are detoxification biomarkers like beta-glucuronidase, and we show you the associated impact of every single biomarker. Longevity factors. This is, in fact, some of the new clinics that we just onboarded are longevity clinics. We know that there's a lot of microorganisms that have, that are able to produce enzymes like myrosinase. Myrosinase cannot be produced by your body. Some of them are found, we do have some levels of myrosinase in your plant foods, but when you cook your food, you tend to break down most of the myrosinase. They become inactive. So what helps you to break down your glucosinolates to things like isothiocyanates? It is your microorganisms. They have myrosinase. And what is the implication of that? These myrosinase have anti-cancer potential. They have antioxidant potential, anti-inflammatory potential, which is fascinating. So is the case with urolithium. We give you insight into lactose and oxalate degraders. And in total, we show you about 30 to 33 biomarkers in the section 3 for the metabolite section. Can everyone still hear me? Because it's raining here.
Yep, we can hear you.
Okay, amazing. Okay, so I'll rush through this quickly. The last stage, the last piece of the test, the section 4, is the recommendation section. So now we've told you what is high, what is low, why you might actually be producing high levels of this metabolite or low levels. The question still goes back to, so what do I do? So this is the recommendation section, the food section. What should I eat? Now we have 5 classes of modifiers. 1, food, prebiotics, probiotics, supplements, and lifestyle changes. The way we have actually built this test, we start with food. What you see in green are the things you should be eating. What you see in orange are the things you should be avoiding. So for this particular individual, and this is as personalized as it gets, because it is based off on what we found in your test that is high and low. So for this individual, they should be eating cranberry, rye, cherry. Why? Because eating cranberry, rye, and cherry will increase akamensia. We saw in their test that they have low levels of akamensia, so we need to increase it. But if you actually want to dig in a little bit further into why we generated that recommendation, you can actually help yourself here and look at the scientific evidence. So 1, 2, 3, which is cranberry, rye, and cherry, those are the academic literature that supports this recommendation. And we do that for every single bacteria that is out of range in your test reports. Why should I be eating sorghum, olive oil, and apple? Because they will reduce bacteroidetes. This person is already high in them. For the avoid section, that is exactly what we're doing too. Ethanol, get away from alcohol, because we have seen academic literature that alcohol actually reduces the amount of akamensia. And that's a beneficial bacteria that helps to contribute to the maintenance of your gut integrity. We just don't do that for food. We do exactly the same for probiotic bacteria. And we show the associated scientific evidence. For supplements, this person should be taking sulforaphane. Why? Because it would reduce bacteroidetes. I think this is one of the ways that we differentiate from, many other known tests out there. One is if you're thinking about gut dysbiosis, if you really want to understand, you know, if you're really interested in your patient's or your client's gut health, you should be thinking about it in the context of gut dysbiosis, not the pathogen deep dive test. This is a test that gives you a true sense of gut dysbiosis versus a pointed pathogen deep dive. But on top of that, it is this recommendation piece where we scour through academic literature, prioritize studies that are clinical studies that were done in humans and well-controlled studies, and we show you all of these recommendations across these 5 different categories. Prebiotics as well. We know that xylo-oligosaccharides would help, and lactulose will help to increase the commensal. We know that things like fructooligosaccharides would help to reduce bacteroidetes. We know that things like chito-oligosaccharides will help to increase rosoboria, which are all beneficial organisms. Okay. That's the last part of the report. But if you're interested in digging more into those references, we show you all of those references here. And this is one of the pieces that practitioners love. It helps us to establish that full transparency, because as you know, the gut microbiome field is an ever-evolving field. As we speak, over 2,000 research papers are produced on the gut microbiome every single month. And so how do we catch up with this? I mean, that's why we are using, AI with some scientists that are on the team to try to capture all of this information. But that's why we also try to show that very transparently in the test report. Okay, I think I'll stop here so that Linta can take over for the sake of time.
Thank you, Chidozie. This is interesting. I learn something new every time I hear it. I am going to share my screen and I'm going to take over by just walking you guys through sort of what are the next steps here? How can you actually access this test? So I've broken it down to the most simple steps that I could think of into 2 main steps. The first one is that you need a practitioner account. You go on our website, which is www.vitrac.ca, and you click on the practitioner tab and you create a practitioner account. Now, your practitioner account is going to allow you to be able to log in and view all your patient results in one place, sort of this convenient portal that is super secure, HIPAA compliant, for you to be able to go back to and review all the results. The second thing that you need is to sign a contract with us, and then what we do, sort of just so we can be really transparent about what the pricing is. And then once you do that, we will give you access to the order sheet where you can go on and start to order tests for your patients. So I explained this a little bit. You go on our website, you click on the practitioner tab up top on the right-hand corner. And then it will take you to this page, which essentially where we ask you a couple of quick questions, your name, your email, you create a password, and then you click submit. And then the one thing that you need to remember here is it will send you an email to confirm your email address, so we can actually make sure it's you. Sometimes that email goes to spam or junk, and in case it does, please check those folders. As soon as you confirm your email, that's it. You can log on and you are good to go. The second thing is signing and returning the contract. So this contract is very simple and essentially outlines that we are going to sell you the test at this price. And that's it. So it's very, very simple. And we just want to be transparent with you and sort of start to work with you in that way. And when we do that, we also offer a quick 15-minute conversation, which will either be with myself or some of my colleagues. And what we'll do is we'll just make sure you're onboarded properly. We have any questions that you have asked answered, and just make sure that you're feeling comfortable and ready to go. And then once that happens, I will send you the order sheet. And the order sheet looks like this. So the rest of the order sheet, there is more to it than just their full name. So you're going to write their name, their address, and your name, so we know who is ordering the test, and you click submit. As soon as you click submit, our team will then ship out the test and your patient will get it. So I want to talk a little bit about pricing here. We've worked really, really hard to make sure that we can keep pricing low and affordable. That is very core to the mission of why we started this company. So our retail price is $210. Anybody can go on our website and purchase a test for themselves. We do have practitioner discount, and this is the pricing that we lay out in the contract I send you, essentially just saying we are going to offer you the test at this discounted price of $175. And then from there, sort of it's up to you to decide if you want to mark it up to the $210 retail price, if you want to bundle other services. We've seen clinics doing a lot of different things. So it's up to the practitioner to sort of decide what works best for their patient. And then we also have bulk pricing. So when clinics start to introduce this test and they know, okay, I'm getting 10, 20, 30 orders a month, whatever it might be. Sometimes they like to switch on over to the bulk pricing, which is sort of the best pricing that we offer, which is $160 per test. These are all Canadian dollars. And the last thing I want to do is sort of just walk you guys through what the whole process looks like from when you order the test for your client and then all the way down to when the results are ready. So the first thing that you're going to do is order, the practitioner can place an order for their client in the order sheet that I just showed you. And when you do that, our team is going to ship out the test to your client's home. Your client is going to receive the test in about 1 to 3 business days. So it's very quick. Once they have the test, they're going to take their sample and send it back to the lab. The client has all of the instructions as to how to collect the sample inside the box itself. So you don't have to sort of walk them through that unless they come to you. So once they ship back the sample, from that point to when it gets to the lab, it can take anywhere from 5 to 10 business days. And then from when the lab arrives at the sample arrives at our lab to when the results are ready can take anywhere between another sort of 10 to 14 days. So the whole process is relatively quick. Within a few weeks, you can be ready to go from when you order the test. And then what happens is you will receive emails throughout the process saying, hey, your client will receive emails telling them your test is shipped out, your test was received at the lab, your results are ready. And then once the results are ready, they will also go into your practitioner portal where you can log on and you can review the results. The last thing I want to point out is we have a very tight-knit community with the practitioners that we work with. So even though we're in hundreds of clinics now, we know all of our practitioners on a name-to-name basis. We know a lot about them. They know a lot about us. We have weekly webinars where we have a standing sort of hour where anyone can drop in and they can bring results. They can discuss cases with us. We can sort of help guide them through how to sort of piece together this puzzle of looking at symptoms and then getting comfortable with, reading and getting acclimated with the results. And all of this is at no cost to the practitioner. It's just a way of us saying, hey, we know the microbiome is complicated. Chidozie has spent over a decade in the lab studying and researching this. So he's sort of the source of truth and is always happy to be there and help our clinicians out. And that's it. Thank you so much, everyone, for your attention. And I think we'll open it up to questions now. Hi, Julia.
Hi, Linta. Thank you very much. You can, stop sharing screen. And hi, Chidozie. I think with your presentation, it probably would have been a good idea to have an artificial intelligence transcriber. You just are, you just are pouring a wealth of knowledge. And I saw people, all the practitioners were staying on, and it's just a lot of information. So I think this is one that we will watch a couple of times, but it is undoubtedly evident the amount of work, but also the amount of interest and passion you have in this subject matter. Right? This isn't just a 1 + 1 = 2. This is something that you're very passionate about.
Thank you very much. Yeah.
Yes. And thank you, Linta, for the letting us know how to actually move through getting the tests. Now I know when you screenshot the folks that are with you on those calls, of course, the top right is Brett Haas. Because it's the first face that I see in there. So it's nice to see him there. I also know Lisa Codispoti and Natalie Ninham. There's a lot of people very interested, but Brett, he's right in there, right? Gut is his game. So it was really nice to have him part of your group. So I love that, because we all really love Brett and we love you too. So we have just a couple of questions. Are you okay with just a few questions?
Oh yeah, absolutely.
Okay. So I'm going to ask, I think they're all from one individual, but does gallbladder removal negatively impact gut microbiota?
Oh, that's a very good question. Yeah, so I think the way you should think about it is, first, to the role of the gallbladder, it helps to store your bile and concentrate your bile for the digestion of fat, right, for lipid metabolism. And so one of the consequences of the removal of the gallbladder, and actually a lot of people who's had their gallbladder removed, one of the some of the most consistent symptoms that they have is, first, changes in bowel habits. They also have issues with bloating and diarrhea. And the reason is because you're just going to be having an increase and release of bile into the small intestine. And not just the small intestine, that reuptake, because your bile has to be taken back again in the distal small intestine. That whole process will be upset. Now, what is the implication of that? Of increased level of bile in your gut? It enriches certain bacteria. And some of those bacteria are actually the bacteria that end up producing gases that can lead to the bloating and changes in intestinal bowel movement. An example of that bacteria is Allistipes. Allistipes feeds literally on bile. Literally feeds on bile. So they are in your colon, but because, and we tend to see this also in people that are a fraction of people that have diarrhea predominant IBS. They have issue with bowel malabsorption. They're not able to because of that bile malabsorption, a lot of bile is getting into the colon, and that's enriching allostipase. That's also enriching bilophila. In fact, the name of the bacteria gives it away. Bilophila, it loves bile, and then that also enriches Bacteroides. The implication here is that all of these organisms then produce high levels of hydrogen sulfide. Hydrogen sulfide is a major culprit in bloating and changes in intestinal bowel movement. So yes, there is quite a direct evidence there. And it completely changes your gut microbial diversity.
Very good. Thank you. Thank you, Chidozi. This might be one for, well, actually for yourself, for Linta. Can we talk about retesting patients? When you do your first test and then our clinicians develop a protocol, what are your suggestions for retesting to see whether we have outcomes or not? What's the period of time that makes sense?
Linta, do you want to go? Or do you want me to stay there? Linta, you're muted, by the way.
You're muted, huh?
Oh, thank you. I would say about 3 months' time is what Chidozi and I typically recommend. And that's one of the really nice things. I think that's been a game changer for clinics, for practitioners, is you can, because the test is now a lot more affordable than other options, you can now run this test for almost all your patients, and you can retest after to see if protocol is actually moving bacteria in the right directions. So we have really interesting stories of practitioners talking to us about, I had tested this one patient, found out that they had absolutely zero Akkermansia, for example. And in that case, particularly, there's no amount of diet that could fix that, right? Like you literally have to reseed it and then feed it. So it's really nice to be able to retest and see if things are moving in the right direction. So we'd recommend about 3 months.
Okay, wonderful. Thank you. Marta has a question. First, she says, hi, and thank you so much, which many of the participants are saying. Just wondering, how is the sample taken, or what type of sample is it that the client is providing? Well,
you want to answer that, Julia? Well, yeah, it is too. So, it's this fecal sample that gives us a sense of what's happening.
That's what it is. I have my happy test right here that I have to do. Yeah. I got 2 of them that we're doing, but it's a stool sample, Marta. Okay, Cameron. I recently found that polysorbate 80 is harmful for the intestinal lining, and have noticed I do not eliminate as often as I used to before this. Any suggestions? Now, that is a personal question. So usually we ask broad questions, but do you have anything to say about polysorbate 80, or is that too granular of a question?
I mean, polysorbate 80 is, I'm guessing she's talking about the non-ionic surfactant, polysorbate. Interestingly, I wouldn't say unfortunately, because this doesn't happen for everybody. Interestingly, it's generally regarded as safe by the FDA for consumption. But I think that this is where you basically just have to listen to your own body. I think one of the biggest pieces of the gut microbiome is what we call bioindividuality, because, Julia and I can have different gut microbiome profiles, but at the same time, we are both healthy. And that bioindividuality is actually what dictates to your body what it can take and what it cannot take. I am a perfect example of that. I am somebody who can't even take alcohol once a month or once in 3 months. It would completely upset my system. I'll be inflamed everywhere. And this is not, once taking alcohol once in a month is probably fine, but not for me. And I think that this is where this whole thing is going. It is personalizing your health, and biohacking starts from listening to your own body. So I think that's what I can say about that. I don't think that there's a one way answer to that. Yeah.
Okay, now I'm going to ask, and thank you, Cameron. I hope that helped a bit. I'm going to ask a specific question. Now, there are gut tests that are out there, and practitioners know what they are. Can you give us the high level, the overview of what makes Vitrax tests different from others? Now, I know that there is a whole bunch of answers to that, but can you give us a bit of an overview as to why should we as practitioners move from other tests and give Vitrax a try, and perhaps even have it as a preferred testing facility?
Yeah, I think it's a fantastic question. I mean, the first reason is, if you are interested in gut dysbiosis, you should be doing a Vitrax test. That's what you should be doing. Because I don't want to mention the names of these other companies, but for a lot of these tests that practitioners use, those tests, the principle upon which those tests operate is they're more like pathogen deep dive tests. So if you're interested in a particular pathogen, you can do those tests to get information about them. But that's actually not how to understand gut health. You understand gut health on a gut-wide scale. I like to tell practitioners, it's not about the presence of the bad guy. A lot of times, it's not just all about the presence of the bad guy. A lot of times, it's the absence of the good guy. And that's why a test like Vitrax is good, because it gives you a total reflection of what's happening there and gives you insight into how the absence or the presence of what they're potentially producing might be causing disease. So Vitract is the good test for gut dysbiosis versus a pathogen deep dive test. The other point is the price point, right? Vitract is probably the most affordable test out there. And the reason that is very personal to us. Now Linta and I even think that in the future it might even be cheaper. It's personal to us because we've had gut health issues and we have had to pay a lot of money through that. So that's the other point. But I think the third point, which is very, very key, is the robustness of the recommendations that we give. Like you take this other test, and this is not to cast aspersions on any other test. Like all these tests are good for different reasons. But in terms of the robustness of the recommendations we give, I would say that Vitract is unmatched in that respect. So these are some of the reasons why I think that when you're thinking about gut dysbiosis, price points, you want to know what you should recommend to your patients, then you should be doing a Vitract test.
Linta, is there anything you want to add to that?
I think that covers it. I think the main thing that we've seen is like practically there are a lot of clinicians and practitioners that we worked with that previously they only had a very small subset of their client pool that could afford to get testing, but now they can run it for almost everyone. They can retest. So it really changes the way you start to work with them, and that can be a big deal.
100%. And I thank you for your diplomacy, Chidozi, because we all know the names of the tests that are out there and they are expensive. And I do have colleagues from IHN who have tried your test, love the test, and they, to me, have been able to recommend and say, yes, Vitract is a test that we are going to move to. And to your point, Linta, we can't afford $400, $500, and $600 for our clients. Our clients can't afford that, right? But they can afford $150, $175, or $200 because you do have to retest.
That's right.
So I like that you're Canadian. I like that you're open to holistic nutritionists. I like the price point, and I like it not as a practitioner myself, but the respected practitioners I know at IHN who are saying, okay, I did a test. I love it. I'm using Vitract. So I'm saying that because it's a brilliant test to use on many different levels, and least of which is the ability for clients to get tests. We can't even afford supplementation anymore because of what is happening in this country. But I don't even want to get off that topic. But so thank you for being reasonable in the price point and just brilliantly in depth in the results. So thank you for that. Let me check any other questions here. Hello, thank you for the wonderful presentation. This test looked very interesting. I particularly love the supplemented dietary section. Do you recommend that we read? Oh, we already answered that, Jessica, if you're still on. It's 3 months after the first test. So I actually think we're good with questions, but now we're going to do the giveaway. So we do have a percentage that we're going to send. It's 45% off of the test, Linta.
$45.
Sorry, $45 off a test. Now I'm going to send that to just everybody who was on the webinar.
Yes.
Right? And you've sent that to me at Julia but we're going to do one test, right?
Mm-hmm. Yeah.
Okay. So let me do the, okay. What I'm doing is I'm going up and down. Oh, and this is for, is it okay for our American students and grads as well?
Uh-huh. Yep.
Yes.
You can send there. Okay. Give me one moment. Okay. Here we go. Olivia Fernandez, you've just won a brand new car. So Olivia, you're still here? Can you let us know? Tuck D's. Yay. Tuck D's. Who is Olivia Fernandez? Are you still here? Because if she's not here, I'm going to draw another one. Hang on a second. Let me see. Beep, beep, beep. Oh yeah, yes, she is. Woohoo! She woohooed up. Okay, very good. Olivia, email Julia at instituteofholisticnutrition.com. And then let me know that you won the test. And then I will introduce you to Linta, and then she's going to get you set up to do that. Okay, so yes, she gave us a big woohoo. Thank you. We're going to package this. We'll get it out to all the practitioners. It'll be up on YouTube. It might give some members myocardial infarction listening to you, Chidozi. I don't think I've ever met a PhD who doesn't speak 100 miles an hour. So I got to go back and just listen to this on slow motion. But it was excellent. So we have practitioners I see who are saying that it was excellent. So obviously that's their bailiwick and they're loving it. Linta, again, thank you so much for getting us through how to actually order. And people will be coming to Vitract, and they can connect with you if they have questions. Yeah.
Mm-hmm.
Okay. And then we'll see a lot of our practitioners out at your Toronto launch event, May 10th.
That's right.
Yeah. And we'll see
and
you
congratulations.
too,
What's that?
and we'll see you too.
Oh, yeah. I'll be there. So congratulations, by the way, on everything that you've done to this point, the launch of the company, all the work that you're doing, and 2024 will be your year. So thank you very much. Thank you for your time this evening. And thank you everyone else who was on the call as well. I know there's lots of things that you could be doing, but you were with us tonight. Okay.
Thank you so much. Thank you for having us.
Very good.
Thank you, everyone.
Okay. Good night.
Bye.